Inside nearly every cell in your body is an instruction manual for how to build and operate you. It's written in a microscopic code that is 3 billion letters long, all tightly coiled into a space smaller than a speck of dust. But how do we actually read a manual that is incredibly long, yet completely invisible to the naked eye? We couldn't just look at it through a microscope. To actually decipher these chemical instructions, scientists had to find a way to take the DNA apart and make those invisible letters glow. The original solution was a technique called Sanger sequencing. This was the method used to read the very first human genetic code. It works by copying the DNA piece by piece, but scientists add a twist: special chemical stop signs. When one of these stop signs attaches to the growing chain, it halts the copying process completely, leaving fragments of varying lengths. By sorting millions of these abruptly stopped fragments by their size, from shortest to longest, researchers can organize the hidden information. Each chemical stop sign gives off a specific fluorescent color. By looking at the colored tags on the sorted fragments, scientists can read the genetic code, color by color, letter by letter. It was incredibly accurate, but because it could only read one small snippet of code at a time in a single straight line, sequencing an entire human genome this way was an agonizingly small task. Using those early methods, the Human Genome Project took 15 years and nearly $3 billion to read just one reference genome. The resulting data filled an entire bookshelf of thick binders. To speed things up, researchers developed a way to multitask on an industrial scale. Instead of reading one letter after another, this new approach reads millions of pieces all at once. The first step is to take that long DNA strand and run it through a molecular shredder, breaking the genome into millions of tiny, manageable fragments. Those shredded fragments are then anchored to a glass slide and copied over and over again, creating localized clusters of identical DNA strands. By intentionally shattering the genome into millions of pieces and grouping them together, scientists set the stage to read the entire code simultaneously, rather than sequentially. Now scientists read the code while they are actually rebuilding it. As each new letter is added to a DNA strand, it lets off a tiny, distinct color flash. A camera photographs these flashes step by step, but this creates a new problem. The raw output is a chaotic avalanche of millions of short, overlapping sequence reads, all generated at once. To make sense of it, powerful computer algorithms act as the ultimate puzzle solvers. They take these millions of tiny, overlapping pieces and match them against a known reference genome, sliding them into their correct order. The bottleneck moved from the lab bench to the computer. The hardest part was no longer getting the data, but processing it at speeds previously thought impossible. This chart shows the impact. The price for a human genome crashed from $3 billion to under $500 today. And if we look at the time required, we see a similar drop, shrinking a 15-year endeavor into a single day's work. Now that it's this cheap and fast, sequencing is no longer a once-in-a-generation milestone. It's a tool doctors can use every single day. One of the most important places this tool is used today is in cancer treatment. Cancer is a disease of the genome. It's driven by uncontrolled cell growth, caused by rogue genetic typos in the body's instruction manual. Today, doctors can take a sample of a patient's tumor and sequence its DNA. By comparing the tumor's code to the patient's healthy DNA, they can hunt down the exact mutation causing the illness. Once they identify that precise typo, oncologists can prescribe targeted therapies that attack the cancer cells directly, minimizing collateral damage. We have come a long way from the painstaking effort of deciphering that first human reference. Today, reading our biological instructions is a routine, life-saving reality in clinics all over the world.